Okay, we have started the stream, everyone. So hello, everyone that is joining us, and welcome to the Paper Trail. My name is Bruna Paulsen. I'm a postdoc at the Gladstone Institutes, and the Paper Trail is an online interactive journal club that was developed by 4 former colleagues turned lifelong friends, myself, Franklin Hall, who is a graduate student at Johns Hopkins University, Dr. Quentin Smith, who is an assistant professor at UC Irvine, and Dr. Michael Blatchley, who is a postdoctoral fellow at CU Boulder. And Michael is going to be your host today. And so, born from our love of discussing science and our yearly paper a day challenge, we came up with the idea of the paper trail to be a platform that allows primary authors to come and share the story of their research. And so we really wanted the paper trail to stand out from other online presentations. And so to do this, we came up with a set of guiding questions that prompts the author to share more than just the results. And so we're so happy that you've decided to join us for this live stream. And in true Twitch form, we ask that you use the chat feature to ask questions throughout the presentation. We have moderators, or the other host will be serving as moderators, and they're going to be in the chat, and we're going to be relaying questions from you guys to Bruna, and Bruna is eager to answer your questions throughout the presentations. So please don't hold them to the end, ask them as they pop into your head. If you are in the stream right now and you don't have access to the chat feature, you can just really quickly create a Twitch login. It just takes a few seconds and you can come back to the stream, and that will give you access to the chat feature. And so with that, I would like to introduce our speaker, Dr. Bruna Paulsen. Bruna Paulsen is a postdoc at the Paola Orlada's lab at Harvard University. Paulsen's research uses human brain organoids to study molecular and cellular bases of autism spectrum disorders. And prior to joining the Orlada lab, Paulsen developed a new strategy to increase precise gene editing in Derek Rossi's lab at the Harvard Medical School and Boston Children's Hospital. During her PhD at the Federal University of Rio de Janeiro in Brazil, Paulsen generated a patient-derived pluripotent stem cells to reveal metabolic and molecular changes associated with schizophrenia. And so with that, I'll let you guys take it away and you can start sharing your slides.
Let's see. Can you see it?
Yep.
Awesome. Thank you so much, Michael, Bria, Quinton, and Franklin for the invitation. I'm very glad to be here talking to all of you guys. And as I told them before, I'm taking this as a challenge because we usually are invited to talk about our work in a more formal way and skipping a lot of steps. And I'm very excited about really like tell you guys in a little bit more informal this work that has been done not just by me, but also my co-authors Silvia Velasco, Martina Pigoni, and Amanda Kedego in Paola Orlada's lab. So I will start today by saying thank you to the people who contributed to this work. And this include all the Orlada lab, especially Paola, and all the support from everybody has been great. It's been long discussions or simply making our days better. As you all know, we spend most of our lives in the lab. So I take this as an important thing. I would also like to give a huge shout out to my great co-authors, as I was saying, Amanda Kedego, which is the bioinformatician that works directly with us, Martina Pigoni, who is now in Switzerland, and Silvia Velasco, who just started her lab in Australia. Also, our collaborators from the bioinformatics side, Joshua Levin and Aviv Regev, along with their groups, especially Shiana Dicons and Sean Simmons. I would like to also say a huge thank to Georgia and Tony, who were actually fundamental, especially in the beginning of this project that I'm about to tell you guys. And of course, all of our great other collaborators who actually brought a lot of different expertise that really allowed us to do this work with using so many different strategies while tackling this very complicated question. So I started putting,
before you move on, we've started to kind of ask people this because I think it's really interesting, and especially when it's such a large group of co-authors on a paper. How did you kind of bring the team together and find these specific collaborators? Because this paper has just so much really cool data in it, and it's really cool to kind of find out how the team came together, I guess.
Yeah, absolutely. So Georgia, Tony, Martina, Sylvia, Amanda, and we all have been postdocs in parallel our Lotus Lab. This project started as 3 different projects at least, and then we saw an opportunity to actually combine them. Joshua Levin and Aviv Regav have been like close collaborators to their Lotta Lab from the bioinformatics side. So I think for pretty much all the projects done in the lab, they are always contributing, and that makes that connection natural. And all the other collaborators that contributed with their specific techniques, and we are going to talk more about each of them, it was more in the need of us doing that specific experiment, and they were the experts in the field. They knew the knowledge, they had developed the technology at times, and then most of them are close by, actually, either at the Broad Institute or MIT, and that's how the group came together. All right, so can I continue?
Yep, go for it.
Right, feel free to interrupt me. So I put this story kind of like in a timeline to give you a sense about the progress of this work until it was published in the beginning of this year, more specifically February. So in 2017, when Georgia was still a postdoc in Paula Lotta's lab, now she has her own lab. She's a professor at USC. And she published this paper, which was, it was the first use of brain organoids in Paula's lab. And in this paper, Georgia showed that she was able to culture these human brain organoids for long-term in culture, which is actually very important because the brain, the way our brain develops, more specifically the cortex, it's in a very long timeline that takes at least 9 months in utero. So you have to some extent, kind of the same amount of time, culture these organoids in culture. So Georgia was able to culture them for 9 months and show that they were recapitulating development. And that was the moment that we already start seeing the potential of these new models to not only recapitulate the human development, but also to study or start studying abnormalities associated to neurodevelopmental disorders. And that was the moment that Georgia was actually the person who, with Paula, had this idea of start using CHD8, which is actually one of the genes that is most replicated and commonly found in genetic studies for autism. But soon after, we were also interested with other collaborators to start discussing about other genes that would be also interesting to be modeled using these organoids. And then in 2019, we were also looking for, we needed actually a very robust and reproducible protocol, which would allow us to understand and better interpret the complex phenotypes that we were starting seeing these organoids. So this work was led by Silvia Velasco, and it was fundamental. It's the basal model that we use in this paper I'm about to present. So, and then when we start getting like all the exciting results that were a major part of this paper, COVID hit, and we were all forced to be home, analyzing all the amount of data that we had generating, and start writing the paper. That was when we made our first submission, which was, you can check that out in the paper, it was in November 2020. And up to now, it was basically the process of reviewing, doing more experiments while a pandemic was happening. And yeah, and that, and the paper was published February, as I was saying. All right, so the way I divided the presentation, I'm going to start with a quick background, and then we are going to come to the first guiding question, as Bria was telling you, that it's what motivated this work. I'm going to show you some of our results, and then we finish with 4 more guiding questions to show you a little bit about the background of what this paper was. So for a very long time, actually, since the Lotta Lab exists, Paolo was very interested, and the lab had been very interested in understanding how the cerebral cortex develops. And this is a particularly interesting region of our brain because it's the region that really computes our most sophisticated functions, such as creativity, movement, language, sensory perception, and several others. But actually, all this complexity relies on the perfect assembling of the cerebral cortex, which takes a very long time. And during this time, each cell type comes in a very specific moment. So in this cartoon that I'm showing to you, it's representing the development of the mouse, the cerebral cortex of the mouse. And you can see that along the time, our diversity of cell types is increasing. So each cell type here is shown by a different color. And in the mouse, this process takes about 20 days until the animal is born. And in humans, the process is fairly similar, but it takes much longer. So for us, our cerebral cortex take at least 40 weeks in utero. 40 weeks since the very beginning of development, but takes all those 9 months for the individual to be developed, for the brain to be developed, and more several years after we are born. And very briefly, just to make sense of the main cell types that we have in the cerebral cortex, we have two major classes of neurons. We have the projection neurons, which are subdivided in multiple different types of excitatory neurons that extend their long axons to different targets in the central nervous system. We also have the interneurons that connect with these excitatory neurons in local circuits, balancing their activity and guaranteeing proper neuronal function. And finally, we have glial cells, which interact with all the neurons, helping refine and support cortical function. And when this process of building the cortex is disrupted somehow, this is when we can lead to neurodevelopmental disorders. And some examples of neurodevelopmental disorders are autism spectrum disorder, intellectual disability, attention deficit disorder, mood disorders, and several others. But altogether, these neurodevelopmental disorders affect about 10% of the population, which is a huge prevalence. And despite this huge prevalence, we still know very little about these conditions. More and more, there are new genetic studies starting, coming up, and each of these genetic studies are pointing to a lot of strong genetic components, such as hundreds of new high-risk genes. But even though it's still very unclear how much this large and heterogeneous collection of genes converge on a common developmental defect. And this is actually due to partially because we don't have a very good model to study human development in the lab. So some of the reasons why we really need models to look at the human neurodevelopment is because the human brain is very different than the other species, for example. But also, most of our development, as I was saying, happens in utero. So we'll never have access to the real tissue, to the actual tissue, beyond very small biopsies in very specific moment in time. So the tissue, access to that tissue is very limited. And finally, and perhaps most importantly, as we learn more about the genetics of these neurodevelopmental disorders, we are constantly reminded about how complex they are, and that they really make them exclusively human. So basically what this is all telling us is that in order to model that kind of genetic contribution, we really need human models. So a few years ago, Silvia Velasco in our lab, it was a paper that described the reproducibility that I showed to you guys in the timeline. She described this new human organoid model that reproducibly generated the cell types of the cortex. So in this image, we see in magenta the progenitors forming these neurotube-like structures that resemble to some extent the formation of the neurotube. And we see in green the first neurons migrating out of these neurotube-like structures. And then if you combine this technology to other technologies such as single-cell RNA sequencing, now we are able to see how reproducible these organoids are. So in this graph, in this figure, what I'm showing to you is here on the left, we have a combination of data from single-cell RNA sequencing of 3 different organoids. And we see each cell type representing the major cell types from the cerebral cortex colored in a different way. And in the right, we have each of these 3 individual organoids. And what we can see is that each of them contribute very similarly to this large compendia of cell types. If we take the same cell line and we differentiate it again in a different batch of differentiation, we get again 3 new individual organoids contributing very similarly to the differentiation of all these different cell types. And finally, if you take a different cell line, of course, in a different differentiation, that different cell line will still contribute in the same way. So, and this model,
real quick, Bruna, sorry, before you go on, what was it about this protocol that led to that reproducibility, especially kind of between the cell lines there? And that's, I mean, definitely an important part of your paper because you use so many different lines.
Exactly.
What was it about this protocol?
Yeah, so the main difference in between this protocol and the previous protocol, actually the previous protocol we call whole brain organoid. It's a protocol that it let the cells differentiate more by themselves. So we give them minimal clues to differentiate. And by default, they tend to make the forebrain. But depending on the cell line, some cells are more biased to differentiate into other tissues. And then in the end, we get a huge variability within the same cell line, but especially if you are using different cell lines. So what this protocol brings to the table, it's basically there's a little bit more guidance during the differentiation process. So we are using 2 small molecules to inhibit Wnt and TGF-beta to kind of guide the cells better towards the neuroectoderm. Okay. So another thing that I was telling you before is that throughout this differentiation process, these organoids also recapitulate very well the orchestrated developmental process that happens in vivo. So since that first work, this is another work that we just put on bioarchives recently by Anna and Amanda, in which a lot of us contribute as well. But we were able to increase the number of time points, number of organoids, cell lines, and single cells. And what we are seeing here, I'm going to highlight a few points to you. At 1 month, these organoids contain still a large proportion of progenitors. So we see that the progenitors here, the major part, most part of the cells, and neurogenesis is just starting to begin. At 3 months, our progenitor diversity starts to increase, and we have different types of progenitors, including autoradioglia, which is a type of progenitor that it's more represented in humans. But we also have a large, higher diversity of neurons generated in these organoids. And finally, up to 6 months, we start having some cell types that are usually present later, such as interneurons and astroglia in these organoids. So they basically demonstrate that the large compendia of cell types are generated in a process that it's very similar to what is happening in vivo. And most importantly, as we were just saying, for every time point, each individual organoid coming from whichever cell line or differentiation batch, they always resemble the counterpart from the similar age, reinforcing how robust and reproducible this protocol is. And this is extremely important because now we have this opportunity to be identifying unbiasedly new developmental effects or molecular or cellular mechanisms that can be affected, especially if you were trying to understand a gene, the importance of a gene in a neurodevelopmental disorder such as autism. So let's go to the first guiding question, which was really, with all this background that we just talked, what motivated this work? So this was the moment that we knew we had access to a model that recapitulates human neurodevelopment, and it's very reproducible. And in parallel, there was an emerging demand to understand the impact of all this new data coming from the genetics field, a lot of new putative genes associated with neurodevelopmental disorders, and how are they really impacting human neurodevelopment? So in Paula's lab, knowing that we had this tool and there was this need, we were always very excited about using our model to start to investigate the molecular mechanisms behind autism spectrum disorder, which is a very early onset disorder. So we thought that the organoids could recapitulate it very well. And it also has a high prevalence in society. So just to have everybody on the same page, autism is a neurodevelopmental disorder. It can be affected by cognitive, motor, sensory deficits. It can be also significantly impacted by social communication, behavioral challenges. And it affects about 1 in 54 individuals. And every day in this country, we have about 195 new cases being diagnosed. And as I was just mentioning, there are more and more new papers coming up pointing to new putative autism-associated genes. This is one of these papers, which is actually one of the largest exome sequencing studies to date. And from the genes identified by this study, the majority of them was expressed since early in development. And they had function in gene regulation, gene expression regulation, or neuronal communication. So we thought that these genes could actually be a great entry point to model this disorder. So basically, we were thinking that if we could mutate them in a control line, we could better understand the consequences of that specific mutation during development. So we decided to ask whether we could use our organoid models to understand whether a specific set of these genes would converge on a shared developmental abnormality. And that paper that I was just telling you, they identified 102 genes, risk genes. And we started focusing these genes into the ones that were significantly implicated in the risk for autism specifically. But they were also predominantly found as de novo protein truncating variants, which are more likely to be associated with the increased autism liability, and that were expressed early in brain development. So we would be more likely to see their effect in the brain organoids. So finally, we selected genes that were from a single functional category, which were the chromatin remodelers. And this is actually a class of genes that are usually affected in autism spectrum disorder. And we know that these genes, they have a very broad function. And we also know that they are widely expressed. So they are expressed at pretty much all the cell types that we have. So this really makes it difficult to predict how mutations in each of them could lead to the disease and if they shared a mechanism that could play a role. What we know to begin with is that these 3 genes that we selected, CHD8, ARID1B, and SUV420H1, they all were associated with severe neurodevelopmental abnormalities. And all of them were also associated with macrocephaly. So we thought that to some extent, they could converge on a similar effect.
Bruna, before you move on, yes. Again, did you consider any other genes? It looks like there are, I think, 100. Oh, it says right there, 102. Yeah. Did you try any others, or did you just kind of have really strict criteria as you just kind of went through, and these worked well, so you didn't necessarily need to, or would you expect some others to be involved in similar ways?
Yeah, we did. So we actually have work currently ongoing in which we are trying other genes. These 3 specifically from the ones that we started are the ones that are involved in chromatin remodelers, and that's the criteria that we took for this paper specifically. But we also have work ongoing with genes involved, other genes also in this study that are involved in other functions. And we are trying to increase, I guess I'm gonna say this the last, but we are trying to increase like the pool of genes associated and see how much they converge, or if we have pockets of genes that are doing like different things. But thank you. Yeah, that's all right. So so to start all this work, we engineered multiple cell lines, all human pluripotent stem cells, controls, And we use CRISPR-Cas9 to induce heterozygous mutation in these specific genes. And then we used our protocol to generate brain organoids, and we profiled them in multiple time points and modalities to try to see developmental abnormalities that could be appearing in vitro. So here are some of the questions that we were interested to begin with. So we wanted to know what cell types were affected, what biological processes were affected, when were they affected, if these alterations converged at all, and if somehow this was dependent on the human genetic background. So I'll highlight some of these findings, and I'm happy to discuss any things that you guys, any question that you guys may have.
Yeah, so when you were kind of deciding which cell lines to use, did you kind of consider the sex and ancestry of the cells, or is this kind of in the future going to consider that and kind of build a bigger set of these lines? And I guess, how did you decide which ones to use?
Yeah, that's actually a great point. So we have a set of lines that we call the psychiatric controls, that they are basically isolated from individuals that they had gone through interviews and they don't have anything in their genetics that put them, that associate their genetics to any type of psychiatric or neurodevelopmental disorder. But we also have a set of other control cells that we know were not affected individuals, but we don't have as much information about their past. The goal now is also, especially now that we are starting to see that the genetic background may play a role in some of the phenotypes we see, we are more interested in opening this and include a large diversity of genetic backgrounds for sure, as well as sex. All right, so one of the first observations that we all had when we were differentiating these organoids, me, Martina, and Sylvia, is that as we repeated these differentiations, what we were seeing is that constantly the mutants seemed bigger than the controls. And I'm showing you here quantification of size for several organoids of multiple batch for one cell line for each of these mutations, SV420H1, ARID1B, and CHD8. And what we see is that the mutants here represented in red, they tend to be always bigger, closer to 1 month in culture compared to the control. And at first glance, we know that it's very hard to compare this macroscopic observation in the organoid with macrocephaly. But the fact that we saw this convergent phenotype, this convergent result in our 3 mutation gave us confidence that we could be seeing something converging among these three mutants compared to the controls. So we started with our first mutation, CHD8, and we developed these organoids. And in about 1 month in culture, which is that moment that neurogenesis is just starting, but most of the cells are still progenitors, we did bulk RNA sequencing. And what we are seeing here is that when we look at the differentially expressed genes in between mutants and controls, we see that a lot of these genes are enriched for terms associated with synapses regulation and organization, axogenesis, suggesting that the beginning of neurogenesis could be impacted somehow in the mutant organoids. We also profiled these organoids by whole proteome mass spectrometry, and we immediately see that some of the proteins that are upregulated in the mutants, they are related to neuronal differentiation, cytoskeleton remodeling, such as beta-tubulin 3, DCX. But interestingly, among the most upregulated proteins, we see DLX6, which is an interneuron fate regulator, suggesting that perhaps we have an early commitment of the progenitors to this interneuron fate at 1 month. And we kept these organoids, all these organoids in culture for a couple more months. We were really waiting for a time point in which we would see a larger diversity of neurons, and that we believe that we could see more relevant phenotype at that first moment. So here we see one example for one cell line, one batch out of all these tries that we did. And we see that the cellular composition of this organoid at 3 months, it shows the diversity of neurons, as we expect, the diversity of progenitors here in green. And when we closely look at the individual organoids from the control or the mutant, we see something actually quite striking. So out of all the cells that we have in these organoids, when we compare, we see that there are these cell populations, the dark blue and the light gray, that are increased in the mutants compared to the control. And those cells are the GABAergic interneurons and the GABAergic progenitors. And this was, we also stained these organoids. Here I'm showing a different staining, that it's DLX2, which is another of these genes associated with interneuron fate. And we see that we have more cells expressing DLX2 in the mutant in the second column compared to the controls. And to better understand the mechanism behind this increase in the number of neurons in the CHD8 mutant, we decided to look at the cells in the developmental dynamics. So basically, we calculated pseudo-time trajectories using Monocle. And then I'm showing you here our results from our beginning analysis. And we see that our cells, when we combine mutant and controls, they basically divide in two partitions. One that goes from progenitors here in green to the different subtypes of excitatory neurons, and the other one that goes from progenitors to our GABAergic neurons. So when we recompute these trajectories just from the partition of interest, the one that leads to our cells that are altered, we plot them in a pseudo-time, we see that towards the end of the pseudo-time trajectory, we have an overrepresentation of the cells of the mutant here in red compared to the cells of the control. You can also observe this in this plot. So what this is basically showing us is that these neurons, they are not just increased in the mutants, but they also seem to be accelerated in development compared to the controls. And this became very interesting when we decided to repeat these experiments in our two other mutations. So because with the CHD8, we already had some suggestion from the bulk results that there was something happening earlier in development, we decided to profile our other mutations since early through single-cell RNA sequencing. So 1 month of differentiation, we were already doing single-cell RNA sequencing of these organoids for our second mutation, ARID1B. So at this stage, 1 month, we usually do not see any GABAergic neurons at all. And for our surprise, when we look individually, again, the controls and the mutants, we see that even this early development, with just 1 month in culture, consistently the mutant organoids have these clusters of cells that are the GABAergic neurons and their progenitors that are constantly presented in all the mutants, but they barely appear in the controls as we expected. And for our surprise, when we do our third mutation, SUV420H1, and here I'm showing you the strongest phenotype that we had with the cell line with the strongest phenotype, we again see this. We see that closely looking, the 3 mutants, most of the cells actually in this case, they are cells, they are GABAergic neurons or GABAergic progenitors. This was very intriguing and exciting at the same time, because we know from prior studies from postmortem ASD or from studies using human iPS cell, patient-derived iPS cells from patients with idiopathic autism, in which it has been suggested dysregulation in GABAergic neurons during development. But that wasn't the only cells that we saw changed in this earlier time point at 1 month. Although these GABAergic neurons were definitely affected, there was also another population of cells, the excitatory neurons, the deep layer projection neurons here in pink. They were also present in different numbers in these organoids. So here I'm showing you again the ARID1B, and we see that this other population is reduced in the mutants compared to the controls. And again, to better understand what was behind this decrease, we applied a similar strategy that we did for the CHD8. So we consider just the cells leading to the differentiation of this specific cell type, this specific neurons, and we place them in a pseudo-time trajectory. And we see again that towards the end of the pseudo-time trajectory, the cells of the mutants in red are less represented compared to the control, as we see here, suggesting that the cell type in the ARID1B is not just reduced, but it's also delayed in development compared to the control. And again, for our surprise again, that very same cell type was in the 1-month-old SUV 420 H1 organoids. But interestingly, at this time, that population here shown in magenta, it was increased in the mutants compared to the control. And when we put them again in a pseudo-time trajectory, we see that they are more represented towards the end point of this trajectory, suggesting that they are increased and accelerated in the case of the SUV 420H1 compared to the ARID1B. So finding the specific alterations in just these two populations of neurons out of all the other cell types was very exciting, because we were seeing something that was consistently appearing. But as I was saying, the same time that we have prior studies suggesting that there are alterations in GABAergic neurons, there is also other studies suggesting possible relationship of deep layer projection neurons with autism. And we know indeed that there is a strong hypothesis of this excitatory inhibitory imbalance associated with autism that could be associated to the phenotypes we were observing. So,
a couple questions. So you said you kind of focus on these two cell types. How did you decide? Was it just that they were different between kind of the control and mutant organoids, or you knew they were going to be important in this, or how did you decide on these two cell types?
It was actually unbiased. We were,
even better.
To begin with, we weren't expecting anything. We were looking at all populations, and those were the two populations that were consistently changed. So yeah, we weren't like specifically looking at them when we started.
Yeah, that's interesting. Yeah, so it seems like the kind of timing of these events are really important. So these populations kind of shift earlier or kind of are downregulated. Is there any kind of importance, or have you started looking at some kind of spatial relationship with any spatial transcriptomics?
That's something we haven't done in this work, but that's something that we are starting to do, to exactly see if there is any relationship, or if there's any meaning about how these cells are.
Gotcha.
So summing up what we just talked until now, I basically showed to you that these three mutations were converging in two things. So in phenotypic abnormalities in just 2 out of several other different subpopulations, looking at them in an unbiased, as I mentioned, and these two cell types were the GABAergic neurons and the deep layer projection neurons. But we also saw that they were doing this by changing the speed of development of these two populations. So this brought us a question, if that was an element of a process, that a convergence process that was obtained by these three mutations through similar convergent mechanisms, or if we were seeing a situation in which these three mutants were converging on this asynchronous development through completely different molecular mechanisms. And this is fundamental, this is important, because this will allow us to understand, when thinking of a therapy in the future for any of this condition, if we should target the molecular mechanism or if we should target the process. So to investigate these possibilities, we start to compare gene expression changes across the mutations and the controls for the 3 ASD risk genes that we had. So we applied the seq2 to calculate the differentially expressed genes, all organoids here that I'm showing to you at 1 month. And we saw that 92 genes were differentially expressed among all these mutants. But actually, out of these 92, just 41 were changed in the same direction, meaning they were either upregulated or downregulated. So out of this 41, 26 were upregulated in all of them. Interestingly, the 26, some of these 26, they are related with neuronal subtype specification or maturations such as SLA, Greek 3, MEF2C, but this is a very low number. And despite this low degree of overlap in differentially expressed genes, the upregulated genes, they still shared enrichment for multiple terms related to neurodevelopment, synaptic structure, or function, suggesting that the genes might not be the same that are being changed, but at least the function that these genes might be having on the cells, these seems to be kind of similar among these 3 mutations. And leveraging our single cell data, we decided to compare cell type specific changes. So for this, we calculated differentially expressed genes in each cell type from control and mutants and crossing among different mutants. So what we see, and we did this for 1 month and for 3 months. So when we compare closely related cells, for example, this green that I'm pointing here is the apical radioglia with the cycling progenitors with this other shade of green, both with SUV mutant, we see that they share a lot of genes that are being changed in between control and mutant. But when we look even for the same cell type, this apical radioglia with SUV mutation, when we compare that with the apical radioglia with the ARID1B, as well as all the other cell types, we don't see the same degree of overlap when we compare one mutation to the other. And this is basically suggesting that although these 3 mutants share some degree of convergence in this neurodevelopmental process, they seem to be affecting largely distinct genes. So the fact that we were seeing this convergence of cell type and in the process, and that this was happening through a largely non-overlapping mechanism, we were very excited and interested about that, puzzled also about that. So this actually suggests to us that we may have to start considering different molecular conceptual framework about how to think about these disorders. So if we want to think about any type of therapeutics in the future, perhaps we should be focusing on shared developmental processes rather than focusing on molecular pathway. And we start also thinking about what would be the long-term consequences of these early molecular changes. So we know that the cortex is made in a very orchestrated and specific way. So if we have cells coming earlier or later than they are supposed to, we start wondering if this could be ultimately resulting in abnormal activity of local circuits. So this is very early days, but we were very excited to do these experiments with Ed Boyden's group at MIT. And what we did here is we performed calcium imaging experiments with them, and we used for that a later time point organoid carrying mutations for SUV420H1. So here we are talking about 4-month-old organoids. And to analyze this spontaneous neuronal activity, we transduced them with adenovirus carrying SOMA-G-CAMP, and then we performed these acute recordings of intracellular calcium dynamics. So yeah, in this video, we are seeing a control organoid and basically showing the activation of these neurons through confocal microscopy. And then when we start comparing controls and mutants, we see that the mutant organoids have reduced frequency and reduced duration of these network bursts, basically suggesting that the early molecular changes that we observe in these organoids, they are somehow affecting long-term function of the neurons. I said it's very early days because the circuits in organoids have to be better characterized and actually have to be better improved. But simply the fact that we see differences, this already suggests that the early molecular changes are having these impacts later on.
Is this type of change in kind of circuit activity, does that also kind of manifest in patients, or is it kind of strictly a developmental difference?
It could be a developmental difference. I don't know results about patient that could be related to that, but that's basically suggesting that function could be altered.
Yeah, okay.
All right. So another thing that is becoming more and more clear in the field is that even these mutations that have a very strong penetrance, they can be shown in different ways in different patients. So we decided to test whether the genomic context by using different cell lines could be actually able to modulate the phenotype that we were seeing in these mutations. So I'm showing you here one example of the SUV420H1 in which we see 3 cell lines that we used. And I'm showing you specifically the phenotype, emphasizing here the phenotype in which we show increase of GABAergic neuron in these 3 cell lines. And what we see is that the increase of the GABAergic neurons, it's always there it's manifested in all the cell lines. But some cell lines, they have that manifestation is strong, while other cell lines have that less strong. So basically suggesting that that effect was dependent on the mutation and the genomic context in which that cell line, that mutation was inserted. So, and this, we observed that it was not only cell line specific, stem cell line specific, but it's also phenotype specific, meaning that for the other phenotype that we saw, such as increase in the deep layer projection neurons in the case of SUV, not necessarily this cell line was the cell line that had it the highest. So I think this information was very exciting in the first moment, and it really like showed to us that for this type of study we really should be using multiple cell lines and trying to investigate. But this is very early days, and I think we still have a lot to understand what is really behind these differences that we see in each cell line.
That's so interesting that you could see such big differences between the cell lines. Is there anything sort of about the characterization of those kind of parental lines that could inform these kind of downstream changes, or is that just sort of not possible at this stage?
So that's something that we did not invest. It was something that we came across and we interpret, and we publish as it is. I think we have to now better understand what is behind what I was saying. But one thing that we observed that might be related or not with that is that when we look at the level of the target gene, so let's say in this case for SUV420H1 in each of the cell lines, the 3 target genes were different depending, the amount of protein was different depending on the cell line. So consequently, when we induced the heterozygous mutant, the level of this protein was reduced in all the cases in the mutant compared to the control, but the level was different depending on the cell line. So this might be behind this difference. It might have nothing to do. So it's something also for future investigation.
Does the source of the cells matter at all, if they're from skin or fat or blood? Is that
I don't know. I wouldn't expect if they were properly reprogrammed to the pluripotent state and epigenetics is as well reprogrammed. I wouldn't expect, but I don't know. I actually, we would have to test that to tell you. All right, so coming to our conclusion and going to our final guiding questions. So this work actually gave us a pretty good sense of how these new organoid models were a suitable model to study complex disorders such as autism spectrum disorder. And we believe that new understanding of multiple disorders, not just autism, but all the others that have a developmental component, will continue to be done and emerging from this type of studies. And hopefully this data will one day inform to development of targeted therapies or understanding of each condition. All right, so I'm ready to move to the last 4 guiding questions, and I can discuss them as much as you guys want. So starting with the method that was critical to complete this paper, here I'm highlighting, I think, the ones that are part of our workflow, which were the CRISPR-Cas9 gene editing, which was something that we had to develop for all the cell lines that we did, the organoid protocol that led not just to this reproducibility, but also to the long-term culture. We had organoids that we profiled up to 6 months, as well as single-cell RNA sequencing, single-cell ATAC-seq, proteomics, and the calcium imaging. But out of all of these techniques, I think most of the work, most of the data that we have come from, of course, the organoids and the single-cell RNA-seq. Those are protocols that were developed in our lab. The single-cell RNA-seq, we have been using the 10x platform, but the dissociation is something that we optimized. And we have both of these protocols well described and posted in this protocol exchange, and you can find it online, in which we detail everything. So if anyone is interested in taking a look, I invite you all to take a look, and feel free to contact me if you have any further questions. So, and for the alternative approaches, I was trying to think of things that could be like more relevant or less relevant. So there are things that are still like in the wish list, but wouldn't have made probably too much the story of the paper. And one of these things is when we have single cell proteomics, that's going to be pretty cool when it's more, but I don't think that would have changed too much the story of the paper in this moment. But one thing that several people have brought up to us in conversation, also some of the reviewers asked us, was the possibility of using patient lines, using patients carrying specifically these mutations. And this is something that we were also interested to begin with. Unfortunately, there are few to any cell lines of patients carrying these specific mutations. It's something that we had been constantly looking, actually, even at Safari, who is making a huge effort right now to generate cell lines from patients carrying several mutations for autism. The thing is that these mutations, the prevalence of each of these mutations is very low. So I had here even one example that in one paper from a cohort of over 6,000 cases analyzed, just 13 had a mutation for CHD8. And to do the type of work that I just presented today, in which we are trying to really have multiple genetic backgrounds and compare these different mutations, it would be probably very hard to, at least nowadays, to have access to this many cell lines derived directly from patients. So the strategy we took was to use mutations in these genes that are strongly associated in control lines, so we could compare the isogenic control. And ideally, we understand the importance of doing these studies with patients, and I think they are going to be complementary to the study we have been doing. But really, to go like in a large scale to test more mutations, we are probably going to have to go with an approach more similar to what I showed you today.
Do most patients have one mutation, or are there patients who have multiple mutations in these?
We try to do some analysis on that as well. It's usually, or at least from the data sets that we analyze, it's one. Okay. They are so rare that I think it would be even more rare to have. All right, and trying to bring to you a little bit about what has changed in this paper since the original submission. So here I brought up a version that we had submitted at bioRxives in November 2020, and what the paper is now. So at that moment, we had 57 organoids, 416,000 single cells. Some of the things that were asked by the reviewers, and with the continuous experiments we were doing that were changed, is in this first version, we had actually compared different genes. So we had SUV420H1, CHD8, and we were comparing P10, which is actually another gene strongly associated, which is not a chromatin modifier. And we decided to, at some point, narrow down into the chromatin modifiers, as P10 started also having some other interesting results. And we hope to wrap up this story soon and have it out, but it made more sense, and also going with some of the comments from the reviewers, to go with defined class of function of these genes, and that's why we focus on chromatin remodelers. So we had one new gene, we had more cell lines. So in the end, combining controls and mutants, we had a total of 20 cell lines in the final version. We had more time points, which was something that the reviewers asked. So in the beginning, we were basically analyzing a one time point per mutation. And then for most of the mutations in the final version, we have at least one data set for 1, 3, and for at least CHD8 6 months. And we also incorporated new techniques. So we did single cell attack-seq to try to understand a little bit the relationship of chromatin accessibility with the data we were getting from the single cell RNA-seq, and as well as calcium imaging to understand the functional consequences of the early changes. And yeah, in the end, the 57 organoids became 112. We had almost 8,000 single cells, so 749, plus 65 organoids that were used for bulk RNA-seq, single cell RNA attack-seq, or proteomics. And I guess this is our last guiding question, which is, what is the next experiment or the final experiment? I think where our group, where Paula's group is leaning to right now, is to try to understand how this convergence expands to other mutation. So we know that there are several other genes that are strongly associated with autism, more chromatin modifiers, but also genes associated with other cellular functions. So how much there's a convergence of all of those genes. And another interesting thing is to better understand this phenotype that we saw in function of these neurons. So how these early developmental abnormalities ultimately impact the properties and function of neural circuits. So I'm happy to expand in any of those. But I guess I will leave with just my contact, a QR code for our paper if you're interested, and a few pictures of Boston that can be completely different depending on the time of the year you visit. But yeah, happy to continue this conversation.
Yeah, wow, amazing work. I think I have a couple minutes left, so we'll maybe just ask one or two more questions, just kind of in terms of future work and that sort of thing. So can this, and maybe it already is, but can this approach kind of be extended to other diseases? I know Bria mentioned you worked with schizophrenia before, or are there other kind of specific diseases that you're interested in looking at, or the group is interested in looking at?
Yeah, so our group is very focused on autism. We are starting to think about other disorders as well. And the reason for autism is the very early onset, as I was saying. These organoids recapitulate very well the beginning of development. So schizophrenia is also known to be a neurodevelopmental disorder, but the onset happens a little later on. It can still be done, and there is indeed a lot of people also considering using these organoids or using these organoids to look at neurodegenerative disorders. We have been focusing on the very early onset ones, but yeah, you could definitely do that.
Cool. I think the group of us have training in bioengineering. So are there kind of other approaches in terms of maturation of these organoids that you are looking at that might kind of help extend that timing from kind of very early stages of development to later?
So yeah, I guess like maturation of cells, I guess several groups, including ours, have been looking. So despite seeing this high reproducibility in the cell types generated, the organization of the cells in these organoids is still not as reproducible. So I think that's something that several groups have been thinking about different strategies to achieve. And it's still unclear if more organized organoid or reproducible organoids in terms of organization will bring any advantage for cell type specification or for neuronal maturation. I guess that's something that has to be done too. Or even like other strategies like vascularization and so on.
Yeah, definitely. So it looks like we have one question from the chat, and I guess we'll kind of finish there. So what would you envision as the long-term implications of this work? In utero genetic testing, therapeutics, or something else?
I think that to begin with, we are trying to understand what is behind autism spectrum disorder. I think the limitation that we have, the lack of models, we not even know what is happening. And in the future, we have to, after really understanding, and this is just the first step, we are far from knowing everything that is behind it. We have to expand this work a lot more. Then we think what makes sense. I don't know if therapeutics will make sense. I don't know if genetic studies, as I think it was mentioned, in utero. Anyway, this, I think it's something to be discussed later on when we have a better understanding of like the full picture, what is behind.
Yeah.
Thank you.
Yeah, thanks for the question. Great. I think we'll wrap up there. Thanks a lot, Bruna, for an amazing presentation.
Thank you so much.
Yeah, thanks everyone for joining the Twitch stream. This video will be posted to YouTube sometime this week. We will tweet about it. So if you're not following us on Twitter, our Twitter is the Paper Trail JC. Yeah, thank you all for joining, and I'm gonna end the stream.